Did you know that the chromosomes commonly seen in the diagrams in these two forms only appear while the cell is undergoing cell division? In fact, while undergoing the early preparation phase for cell division, which is denoted by G1 in the cell cycle, and the final preparation phase for cell division, which is denoted by G2 in the cell cycle, chromosomes appear as thread-like structures that will only become visible as the cells begin to divide in the M phase, but not while it is preparing to divide, as in G1 and G2, which are part of the interphase of the cell cycle. In each of the chromosome, DNA is associated with protein molecules that ensures that the enormously long DNA molecules are being physically organized in a manner that ensures that the development and maintenance of even the simplest organism can be contained within the tiny cell space. Each DNA molecule itself consists of two complementary chains of polynucleotides, with each strand consisting of 4 types of nucleotide subunits. They each consist of a 5-carbon sugar, which is of the deoxyribose type, to which a phosphate group is attached, and a nitrogenous base, or nitrogen-containing base, which can either be adenine, guanine, thymine, or cytosine. Each nucleotide is composed of a sugar phosphate covalently linked to a base, guanine in a given example. The nucleotides are covalently linked together in a chain via a sugar of one nucleotide molecule to the next via the phosphodiester bonds, linking the 5' end of one sugar to the 3' end of the other sugar. Each nucleotide molecule is therefore imagined to have a phosphate knob, indicated by the 5' phosphate, and a hydroxyl hole, indicated by the 3' hydroxyl, so that the DNA strand with a sugar phosphate backbone, from where the bases adenine, cytosine, guanine, and thymine extend from, is the result of the interlocking of these knobs and holes. For a DNA molecule to consist of two DNA strands held together by hydrogen bonds between the paired bases, it is necessary for the two strands to run anti-parallel to each other and be oriented with opposite polarities, because otherwise they will have all of its subunits lined up in the same orientation. The two ends of the strands can easily be distinguished, as one will have a hole denoting the 3' hydroxyl and the other a knob denoting the 5' phosphate. While the two DNA strands that lie anti-parallel as shown here are being straightened out, the reality is that they wound into a double helix. All the bases are on the inside of the double helix, and that there are 10 base pairs per helical turn. The sugar phosphate backbones are on the outside.